Every cell in the human body operates using biological data. Inside the nucleus is a chemical instruction manual that governs how we function. This double helix structure contains thousands of specific segments called genes. These function as individual lines of biological code. These genes determine physical traits, such as eye color and height, by providing the exact specifications for building and maintaining the body. However, this code is not always copied perfectly. Molecular errors can occur during replication, creating a typo within the manual. These errors are passed down through generations. They are the underlying cause of hereditary conditions like thalassemia, cystic fibrosis, and Huntington's disease. For centuries, medical intervention was reactive. Doctors waited for the body to show signs of failure before using generalized treatments to manage the resulting symptoms. Successfully managing a genetic disease often depended on chance. We lacked the technical means to analyze the operating manual before a health crisis occurred. Today, modern laboratories across the world can now sequence and analyze this complex chemical code with incredibly high precision. Using blood samples, genetic testing allows scientists to scan a patient's DNA for specific mutations that indicate a predisposition to disease. This data functions as an early warning system, identifying the statistical likelihood of developing conditions like heart disease, diabetes, or certain cancers. Patients use this information to pursue early interventions and lifestyle adjustments, addressing risks before they manifest as physical illness. The next phase involves interpreting this data to create personalized medicine, treatments tailored to the individual's unique biology. This precision model allows doctors to choose therapies based on the specific molecular profile of a patient's disease. This targeted approach is used in oncology to treat tumors. Doctors analyze the cancer's genetic makeup to select drugs interacting only with mutated cells. This specificity increases treatment effectiveness, while reducing the impact on healthy tissue and minimizing side effects. By reading and interpreting the genetic code, healthcare is shifting from generalized guesswork toward microscopic accuracy. Today, medical science is now moving from the mere observation of genetic code to the active repair of it. A technology called CRISPR gene editing is making this transition possible. CRISPR functions as a molecular tool. It is programmed to navigate a DNA strand and identify specific damaged sequences. Once the target is found, the tool removes the genetic error and replaces it with a corrected sequence, repairing the strand at the molecular level. This capability allows medical science to address the genetic origin of a disease, to stop symptoms before they begin. CRISPR provides a method to remove inherited flaws from a biological lineage, addressing the root cause of the condition. Healthcare is transitioning into a proactive era. Medical practice is becoming more targeted, focusing on precision interventions at the molecular level. This technology utilizes genetic code, which is highly personal data. As these tools become more common, maintaining the security and ethical use of this information is a central requirement for the medical community. When managed correctly, these advancements provide a path toward reducing the global impact of hereditary diseases and improving long-term health. Our understanding of DNA is no longer limited to studying the history of our evolution. Utilizing these tools gives us the ability to address inherited conditions at their biological source.